Welcome back, and this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and let's continue our discussion about the reproduction in organisms. So, we will continue with the asexual reproduction, and the topic what we are going to cover within the asexual reproductions are so, we are going to discuss about the binary division. We are going to talk about diffusion, then we are going to talk about the budding, then we are going to talk about the spore formation, and at the end we are going to talk about the vegetative reproduction or vegetative propagation. As we discussed earlier also, the vegetative propagation could be of two types. It could be the natural, which is actually happening in the nature, or it is happening without any kind of manipulation done by the human being, or it could be the artificial. And while discussing each of these different methods of asexual reproduction, we will take a couple of examples to understand how the same process has been modified in different organisms to suit to their life cycle. So, let us start with the binary division. So, in the case of binary division, it is actually, or it is also known as the binary fission. It is actually like dividing the cell into two. So, in the binary fission, what happens is that you have a parent cell which is contain a nucleus, okay, and then it gets divides into two. So, there is a clear cut division of the cell by a mechanism, and then it actually forms the two cells, okay, and these are called as daughter cells. We have couple of examples or couple of organisms in which the binary fission is happening. The most probably is the binary fission is mainly been predominant mode of asexual reproduction in the prokaryotes as well as the lower eukaryotes. So, let us take an example of the simplest prokaryote organism, that is the bacteria. So, how the binary fission happens in bacteria? So, in bacteria, so you know that bacteria contains a cell, okay, which is contain a cell wall, right, and within the cell wall you have the cytosol, okay, and then it is having a chromosome which is attached to the cell body. So, this is the parent cell, and in the case of bacteria it involves many steps. So, what happens is in the first step this cells actually grows, okay, and this the chromosome also replicates and grows larger, okay, and then the cell is getting transversely divided into two parts. Okay. So, then this furrow is going to advance further. So, it is going to form like this. Okay. And you will see that one part is containing one part of the chromosome, the other part is containing other part of the chromosome, and eventually this is going to be dissolved and you are going to have two cells each containing its own chromosome. So, these are the daughter cells, okay, and these are the parent cells. So, by making a one binary fission, the bacteria is going to divide into two. So, and this and that is why if you and under the favorable conditions, for example, in the case of E. coli, this binary fission completes within the 20 minutes. Okay. So, if you want to divide, if you want to calculate the number of bacteria which are present after 1 hour, then what you can do is, since in 20 minutes it is going to be double, you can calculate how many doubling cycle is going to happen. So, let us take an example how the binary fission actually increases the number of bacteria in a given time. So, as we discussed, for E. coli it takes 20 minutes for one binary fission. This means in 20 minutes one bacteria will get converted into two bacteria. So, this means, and imagine that you start with 1000 bacteria and I will ask you what will be the number of bacteria after 1 hour. So, if you start with 1000 bacteria, in first 20 minutes it is going to be 2000 bacteria. In another 20 minutes it is going to be 4000 bacteria, and then after another 20 minutes it is going to be 8000 bacteria. So, you can see that in 1 hour you are going to have 3 such cycle, and because of that 1000 bacteria is getting converted into 8000 bacteria, means in 1 hour the bacteria number is going by 8-fold. So, that is the power of the binary division. Okay. Now, we will take another example, and by taking that another example which is from the eukaryotes, we will understand how many different ways in which the binary fission can be performed. So, the simplest eukaryotic organism is the protozoans, okay, where you have the amoeba and other organisms. So, in the case of protozoans, they have they also replicate or divide, they also reproduce by the binary fission, and they have 3 mode of binary fission. One is called irregular binary fission. Okay. So, irregular binary fission normally happens in amoeba. Okay. So, you know that amoeba is a randomly structured organism. It does not have its usual structures, okay, and it has a nucleus in the middle, and then what happens is this nucleus actually grows and grow in size. Okay. So, there will be a synthesis of nucleic acid, okay, and then this amoeba will going to be dissected by a furrow into the multiple steps. Okay. So, this ultimately what will happen, once this will going to happen, then this will be like this. Okay. So, there will be a furrow which is dividing the amoeba this way, and then eventually you will have 2 amoeba, both of them containing their own nucleus. So, why this is called as irregular binary fission? Because this plane of division can happen at on any angle, like it can happen this way, it can happen this way, it can happen this way. There is no way that it can be regulated. The only difference is that it is always been perpendicular or right angle to the growth of the nucleus. So, once the nucleus grows in this direction, it will be right angle to the nucleus, so that it divides the nucleus as well as the cellular content into the 2 equal halves, and that is how you are going to have 2 amoeboids. Now, the second mode of the binary fission is called as the longitudinal and the longitudinal mode of binary fission is happening in the another class of protozoan organisms known as in the Euglena. So, in the case of Euglena, Euglena is a leaf like structure, okay, and it contains a cilia on the top, and then it contains the nucleus, okay, and then the Euglena actually divides or grows in this in the longitudinal direction, and ultimately in the first step what happen is you are going to have 2 Euglena like this, okay, and this furrow will increase, and then you will see that nucleus is also growing, okay, and ultimately this will grow, and you will going to have 2 okay. So, in this case this is a longitudinal division, means the nucleus is growing and it is getting cut from the top, and that is how you are going to have the 2 Euglena on the both side. One is this, another one this one. Okay. The third mode is called as transverse binary fission, and that transverse binary fission is happening in another class of protozoan molecule, and that is known as the paramecium. This is the structure of paramecium, and what you will see is a nucleus and containing the cilia, okay. And so, in this case the paramecium replicates in a transverse mode, okay. So, it grows in this direction, and that is how you are going to have 2 paramecium growing in this direction, and its nucleus is also growing, okay, and then eventually it is going to cut in the middle, and that is how you are going to have the so this is a like this, and then ultimately you are going to have the 2 paramecium like this, okay. So, in this case you are actually cutting the paramecium from the middle, and that is always going to be from the middle through the transverse section. So, the nucleus will grow, then the paramecium will also grow, and it will grow and have the whole body part increasing on both the sides, and then transversely it was going to be divided into the 2 individual daughters. There is another variation of transverse binary fission which is happening in the another organisms known as the planaria. So, in the case of planaria, so planaria is a leaf like organisms. So, what happen is it actually contains a cell body. Okay. So, this is the structure of planaria. Okay. So, where you this is the mouth part and this is the base part, and then when the planaria has to go for the transfer binary fission, it actually goes to any solid surface and then stick to that. Okay. So, by sticking to the lower portion, and then the planaria actually move its body in the forward direction. So, when it stretch its body in the forward direction, what happen is that this portion goes like this, and then it goes, and then so because of this stretching, it gets thin in the middle, and eventually this furrow advance further, and you are going to have a planaria which is containing a mouth in the front, and you are going to have the another planaria which is attached to the protum, does not have a mouth. Okay. So, this portion actually develops the tail and eventually becomes the daughter planaria, and this portion develops the head. So, this portion develops head, this portion develops tail part, and eventually they all two becomes the two individual planaria molecules. And that is why, because of this unique mode of the binary fission, the binary fission is always been considered that the molecule which or the organisms which replicates through binary fissions are considered to be immortal, because if you see even in the case of planaria, this planaria molecule, which is actually the parent molecule, is not dying, because it is just changing its form. Now, it is a parent molecule, it is dividing himself into the two parts. The lower portion is going to be the daughter cell, but the top portion is still be the same planaria what it was there, except that its cell mass or the body volume is lower, and that is why most of these molecules are considered to be immortal and they do not die, because the original parent molecule remains throughout as long as the organism is surviving. Now, we will talk about the second mode of the division, and that is called as multiple fission. So, what happened in the multiple fission and how it is different from the binary fission? In the multiple fission, and the parent cell is dividing into the multiple daughter cells, and so what happen is, so this is your parent cell, okay, and then this parent cell is dividing into the and it contains a nucleus, okay, and then this nucleus is dividing into the multiple nucleus, okay, and then these multiple nucleus are forming the individual cells, and these individual cells are still being enclosed within the parent cell, and then they are being released by the rupturing of original wall, and they releases the multiple cells at the same time, okay, each containing their own nucleus. So, there will be no division of parent cell, it is actually the growth of the nucleus, and then that individual nucleus is getting its own cellular content, and then ultimately all these daughter cells are being released by the rupturing of the original plasma membrane. We have two examples under this category. One is the example of the malaria parasite. So, I am sure you all know about the malaria parasite, but even then I thought we will briefly discuss how it actually do the replication or complete its life cycle. So, malaria parasite actually grows into the RBCs, and it has 4 distinct stages within the RBC. So, it starts with your normal RBC, and then the normal RBC is getting infected by the merozoites, okay, and it forms the first stage, and that is called as ring stage. This ring stage then, so the ring stage containing parasites then eats up the nucleus, and then ultimately get converted into the trophozoites, and that trophozoite further grows and form the zygont, okay. And then this zygont actually releases the merozoites, okay, and that merozoite actually infects the new RBCs, and that is how this malaria parasite replicate within the human blood and completes its life cycle. So, this is the step, the conversion of zygont to the merozoite is the step at which the multiple fission is happening. So, in this case what happen is that once the malaria parasite reaches to the zygont stage, so you can imagine that this is the zygont containing cell. This divides and forms the multiple merozoites, which are enclosed within the original periphery, and then because these merozoites actually exerts the pressure, this get blast, this actually lies the RBCs, and all these merozoites get released into the blood to infect the new RBCs. So, these are merozoites, okay, and this is called as multiple fission. The second class of multiple fission is also happening in the case of amoeba. So, in the case of amoeba, we have a cell which contains the nucleus, and this nucleus divides into the daughter nuclei. So, what we have is we have a cell, this contains nucleus. So, this is amoeba, okay, and in this case this nucleus divides into the multiple nucleus. So, these are the individual nucleus, this is your parent nucleus, and then this individual nucleus forms their own plasma membrane and their own plasma membrane. So, that can get converted into this. This kind of small is formed, and then this particular structure is getting another thick coating to protect it from the environmental stress, and this thick coating is known as the cyst. So, all these are going to be within this thick coat, which is called as the and this structure is known as cyst. So, under the adverse conditions, the amoeba actually forms these cyst like structures, and they are also known as they are also sometimes known as spores, and then this the original cell wall splits. So, within the one cyst what you have is the nucleus, you have the cytosol, and you have the coat, the hard coat which is forming the cyst, and this cyst is withstanding the extreme environmental stress, and this gets dispersed. So, individual cyst is going to be dispersed once the original plasma membrane is going to be disrupted, and they will remain quiescent until the extreme condition is existing. And once the existing extreme conditions are over, then this individual cyst is going to release the amoeba. Okay. Individual cyst is going to release the amoeba, and that amoeba will grow. So, this kind of formation of this kind of structures are always been favored to protect the organisms from the different types of environmental stresses. Now, we will talk about the third mode of asexual reproduction, and that is called as the budding. So far what we have discussed is that if a small the cell body is getting divided into the multiple structures, but the parent molecule is also getting converted. So, now the third mode is called as budding. So, in the case of budding, what you have is you have a parent cell, okay, and that contains nucleus. This parent cell then actually elongates a small portion of its cell body, okay, and then eventually this elongated portion is becoming the daughter cell, and this becomes the parent cell. So, in this mode you have a small projection coming from the parent cell, and then this small projection get enclosed here, and then that is how it becomes the smaller portion becomes the daughter cell and the larger portion becomes the parent cell. We have two examples, and this kind of budding is very common in most of the fungi as well as the algal species, and we have two examples in this class. One is called the yeast. So, in the case of yeast, we have a parent cell, okay, and that contains the nucleus, and then a small portion develops into a bud like structure, and then the nuclear content is also being divided between the parent cell as well as the daughter cell, and then the both parent cell as well as the daughter cell develop into the individual organisms. Then we have another example of that is called hydra. So, hydra is an organism belonging to coelentrates, and in the hydra what you have is actually the cell body, okay, and on the top of cell body you have this stem like structure, okay. So, and the lower portion is attached to a spectrum, or sometimes it is free. So, what happen is whenever the hydra need to grow or do asexual reproduction through budding, a small bud comes out from this lower portion, okay, and this is the parent structure, okay, which contains hydra, right, and then there is a small buds comes out from the cell body, and these small buds then eventually develops into the new hydra, okay, and then after some time this cell body also get broken and develops into the new hydra. So, this is the daughter hydra and this is the parent hydra. Just now what we have discussed about the amoeba, that it actually under the adverse condition by doing the multiple fission, and then all these multiple fission it is protecting that by forming the cyst. The same thing is happening in other class of organism, and they form these spores, and that is another way of the asexual reproduction. So, after this let us discuss the another method of asexual reproduction. So, as you know that sexual reproduction actually requires the lot of nutrition, okay, because the cell has to grow and then it has to be divided, as we have discussed for the multiple fission as well as for the binary fission. Okay. But in many cases what happen is that the nutrition or the environmental conditions are not so favorable, and under those conditions every organism wants to continue its life even during that during the harsh conditions, and in that case it goes with the another mode of asexual reproduction, and that is known as the spore formation. So, in the spore formation what the organism is going to do is it is going to produce the spores, and these spores have are going to withstand the nutritional requirement, it is going to withstand the environmental stresses, it is going to withstand different types of harsh conditions like temperature, humidity and so many other parameters. So, that is what we are going to discuss now. So, the next method is the spore formation, and the spore formation is always been governed by the condition that it is not it is going to an organism will go for the spore formation only when there is a requirement for to withstand the harsh conditions. So, with the spore formation it is going to tackle three problems. One, it is going to tackle the environmental stresses like temperature, humidity, okay. And number two, it is going to use, since these once the organism start producing these spores are sometimes are very light or sometimes they have the organelles so that they can swim or even they can go along with the air. So, that also allows the organism to disperse from one place to another place. So, this is also being used for dispersal, okay, means distribution from one place to other place, okay. There are many different types of. So, in a generalized scheme what is happening, that you can imagine that this is an organism, okay. So, it will form many types, multiple spores, okay. And then these spores, once these spores will get going to be distributed into the environment, and in general these spores are resistant towards the environmental condition, and sometimes they are also being equipped with the machinery so that they can go from one place to another place. So, now we will discuss what are the different types of spores are being formed. So, spore formation is a very popular method of asexual reproduction in the prokaryotes as well as the lower eukaryotes. So, the first spore is known as zoospore, okay, and the zoospores are being formed within a specialized machinery that is called as zoosporangia. So, what is zoosporangia? Zoosporangia is like a double walled bag like structures, okay, and the spores are being developed within this, okay, and then zoosporangia is having a opening on the top, and through this these spores go out, okay, and most of the zoospores are having the very thick cell wall, and they also contain the flagella. Okay. So, they contains the flagella, okay, and they have a very thick cell wall. So, because of this these two character, the zoospores are acquiring the resistance against the environmental stress, and as well as because they have the flagella, these flagella allows them to swim from one place to another place. So, the fungi in which the zoosporangia is developed, the zoospores are being dispersed into the environment, and then through the water they go from one place to another place. The class of fungi in which the zoosporangia is formed is known as the Phycomycetes. The examples in this class is the Eclaima. So, and then we have another class of spore, that is sporangiospore. Okay. So, sporangiophores are the spores which are non-motile. So, they do not contain the flagella like apparatus, and they are formed in a cell body or in a defined structure that is called as sporangia. Okay. So, you can imagine that there is a cell body, and then this is the sporangia in which the spores are formed, and then this break open from the top, and that is how they start dispersing. Okay. But the compared to zoospore, the sporangiospores are very light. So, they do not and since they do not have a flagella, their dispersal is mostly by the air. So, they spread into the air, and then they are actually goes from one place to another place. And another difference between the zoospore and the sporangiospore is that they are mainly being meant for not to withstand the environmental stress, but to go from one place to another place, means they are actually being used to spread the particular organisms in the environment. And the classical example of the fungus which is actually forming the zoosporangiospore is the Rhizopus. Okay. Then we have third class of spore, that is called chlamydospores, and these chlamydospores are the thick walled spores, and they are being developed thick walled spores, and they are the resting spores produced directly within the fungal hyphae, and they are resistant for the environmental stress. So, you can imagine that they do not need any specialized the instrument. Okay. So, within that fungal hyphae, these spores are being formed. So, these are the spores, and along with the hyphae, they get dispersed from one place to another place, and these spores are of very thick walled. Then we have another series of spores, that is called oidea. Okay. So, as you know that in the case of chlamydospores, the spores are formed within the fungal hyphae. In this case, what happened is that the hyphae cells. So, you can imagine that you have the fungal hyphae, and that contains different cells, and each of these cells actually get converted into a fungal spores. So, in this case what will happen is that the terminal part of the terminal part of the hyphae will get converted into the spore, and then this get broken, okay, and that is how this will get dispersed into the environment, and they cannot survive the. So, they can be these oidea also can be used to grow, to allow the propagation of organism from one place to another place, but these oidea are of thin walled, and so they cannot withstand the environmental stress. And then we have the second and last class of spore formation, that is called conidia formation. So, these are the non-motile spores, and they are actually being formed either the single spore or a chain of spores by the constriction of the tip of the fungal hyphae, and they are dispersed by the wind or and they will germinate to give rise a new individual. So, you can imagine that you have a cell body, and then the cell body is getting terminated into. So, individual hyphaes are getting converted into the conidia, and these conidia are then dispersed by the wind. So, they go and detach from the main hyphae, and then they get they develop the new fungal cell. So, apart from the spore formation, the another mode of asexual reproduction is the vegetative reproduction. So, the three asexual reproduction mode, or the four what we have discussed, this means the budding, binary fission, multiple fission, and as well as the spore formation is more common in the lower in the prokaryotes as well as the lower eukaryote, whereas the vegetative propagation is more common in terms of the lower plants as well as the higher plant species. So, you can know that the vegetative propagation means what is mean by vegetative propagation? Vegetative propagation means that that every plant has three main parts. What are these parts? A plant has root, a plant has stem, and a plant has leaves, okay? And all these parts are known as the vegetative part of the plant, okay? So, all these vegetative parts can give rise to the new plant, and that is what is known as vegetative propagation. Apart from these vegetative parts, you also have the flower, you also have the seed, okay, but seed does not come under the vegetative propagation, because seed is actually the result of the sexual reproduction between a male and a female plant to form a seed, and that seed actually is not the vegetative part. So, since we have three different vegetative parts, all these three parts can have the multiple ways in which they can actually give rise to the new individual. So, let us start with the part number 1. So, in the case of root, so So in all these three methods, the particular vegetative part actually develops the buds. So in the case of root, what you can see is that a root actually is having the buds, okay, and these buds actually, when you take this particular vegetative root and put them into the stem, into the soil, or if you suppose you cut these into the multiple portions, multiple multiple pieces, okay, and then if you take the individual piece which contains these buds, okay, and if you put them into the soil, the bud actually develops the root on one side, okay, and it develops the shoots on the other side, okay. So this is the shoot part, and these are actually the buds which are present, and then this is the root part, okay, and eventually this will develop into the new plants, okay. And the classical example of the plants which can be propagated by the vegetative mode is the sweet potato. And you might have noticed that in the sweet potato you might have seen these bud like structures. So, if you cut any sweet potato in such a way that you do not damages these buds and you put them into the soil, that each bud will give rise the new plant. And then we come to the second part, and the second part is the stem, okay. You can have the stems of three different conditions. Number one, the stem which is actually below the ground, okay. So, you can have the stem which is the underground. So, within the underground stem you can have the multiple ways in which, or multiple structures which are actually being found in different examples, and they are actually known to form the different types of plants. And what are these variations? So, in the first is it is called suckers. So, these are the stems which grows below the stem, below the ground, okay. So, you can imagine that you have this shoot, okay, and this is the root, okay, but in this suckers, this particular plant has a stem which goes along the, so this is the soil actually, okay. So, it goes along with this, and then it has the buds, and along this bud another shoot comes on the top and the root comes at the bottom, okay. So, this becomes the new plant, and that is, and this mode of doing the vegetative propagation is known as the suckers. The classical example is mint, or the in common name we known as the pudina. And then we have rhizomes. The rhizomes are also the different class of stems, and they are present below the ground, and they also have the buds, and these buds actually eventually develop into the shoot on the top and the roots at the bottom. The examples of one of the rhizome is ginger. So, the ginger what you consume, you can use that ginger keeping the eyes or keeping the buds intact, and then you put them into the soil, and that will give you the new plants. Then we have the corns. Okay. So, corns are actually the highly condensed stems, and they also have the buds, and they are actually present below the soil, and they and in this particular stem you get the buds get activated, and these buds give rise to the new plant. And the examples of corn is the banana. Then what you have, apart from the corn, you also have the bulbs. So, in the bulb, these are also the modified stems which are present in few class of plants, and these bulbs can give rise to the new plant, and the examples in the bulb is the garlic, onion, and as well as the lilies. Then it comes the tubers. Okay. So, the tubers are actually the stems which actually get modified, and then they actually have the several buds, and the tubers are actually the stems which actually stores very large quantity of food material. So, because of that, the stems get swell into a balloon like structures, and then it contains the different types of buds on its body. Okay. So, these are the buds, and if you take these buds and you put it into the soil, what you will happen is the top portion it will develop the shoot, and the bottom portion it will develop the root, and the classical example of tuber is the potato. You can also have the stem which is growing on the cells on the soil surface. Okay, and these are called as creeping stems. So, in the creeping stems, what happen is that the stem is running along the surface of the soil, and then it actually give rise to the different types of plantlets, and there are three variations in this. One is called the runners. So, runners are actually the same as suckers, except that they do not go within the soil. Okay. So, what happen is you have one plant running like this, and then this is the surface. Okay. And then there is a one shoot which goes on the top, and then after some time the buds get activated, and then they form the shoots on the top, and they form the roots at the bottom. So, if you cut from here, okay, this portion is going to be the new plant, and this mode of forming the new plant is called as the runner. Similarly, you also have the stolons. So, stolons are similar to runners, except that they do not grow, they do not. So, stolons are actually can run and can cross even the smaller obstacles, like if there is a, suppose if there is a some hill or some stone. So, in those cases the runners will not be able to cross the stone. Okay. But in the case of stolons, they will actually be able to cross these kind of small obstructions, and the classical example of stolon is the strawberry. And in the case of runners, the examples is the oxalis. Similarly, we have the third variations of the creeping stems, and that is called offsets. Offsets are actually running between the nodes, and they are like runners, but they run with the one node at a time, and then they develop the leaf leaves or the shoot at their budding point, and the examples of offset is the pistia. Okay. So, apart from that, you can also have the stems which is running on the surface, and those are called aerial stems. Okay. So, aerial stem is actually the usual form of stem. So, you can also have the aerial stems. So, in the case of aerial stem, what happen is, suppose this is what one stem, okay, this is the soil, okay, and this is the root. Okay. So, what happen is this portion, so this is the stem, okay, so this is the stem. What happen is this portion get broken and get into the onto the soil. Okay. So, eventually it develops the roots at the bottom, and it actually forms the leaves and other kind of items on the top, and that actually is the aerial stems, and the classical example is the cactus. Okay. Now, after this we will also going to discuss about the leaves. So, in the case of leaves, we have leaves which actually are developing the buds on their surface, and these bud containing leaves are actually causing the development of shoot on the top and the roots at the bottom. So, you can imagine that you have a leaf. Okay. So, at every edges point, okay, so this is connected, right, this is connected like this. So, on this you can imagine that you here there is a bud, and then this bud actually develops into the shoots on the top, and this buds also develops the root at the bottom, and this particular portion, if you put it into the soil, that is going to give you the new plant. And the classical example of leaves as a vegetative organ is known as bryophyllum. Okay. What are these artificial method? Artificial method means the method where the human intervention is required. These methods are cutting, these methods are layering, these methods are grafting, number 4. And then we have the micropropagation or the plant tissue culture. Okay. So, all these we are going to discuss in the subsequent lecture. Thank you.